can stain the cells with different colors as well. So it's a beautiful, beautiful picture. Almost like the starry sky at night.
Fellow Homo sapiens, welcome to or welcome back to Epilepsy Sparks Insights. Now, still 30% of people with an epilepsy are not able to control their seizures with the medications provided. And too many people who do have their seizures controlled with these medications experience awful side effects. Plus, then there are the morbidities like depression, anxieties, movement disorders, cognitive deficits, so many different things, which can be equally, if not even more, negatively impactful upon a person's life than seizures. So that is why we need researchers to help us find something better. Now, most of us have heard, I think, about stem cell research. But did you know that we have scientists studying human organoid cells, which are derived from our stem cells, to work on improving treatment outcomes for people with an epilepsy? And this is what we call working on personalised medicine. So I am excited to be chatting today with consultant neurologist and researcher Patrick Kwan from Monash Uni in Melbourne, who can share with us a snippet of his research into the aforementioned.
So hi, Torie, and nice to meet you and nice to see everyone. So I'm an adult neurologist. I guess my day job is to treat patients with epilepsy, but I'm also a researcher. I'm a professor at Monash University. And I also where I do a lot of research into finding ways to improve the outcomes of people with epilepsy using a variety of techniques from both in laboratory based, but also looking at clinical data and even looking at health services, health economics. My other hat is I'm the co-director for Monash Institute of Medical Engineering, which is a platform within Monash University to promote and educate and train people to do research to bring medical technology solutions for unmet clinical needs. So it brings together 4 different faculties: medicine, engineering, IT, and art and design.
Art and design, that's cool. Well, I guess that is an important part when designing something, isn't it? It's got to look swish enough and function. So you're doing some work, well, you've got multiple projects, but you're doing some work with MRFF grant into stem cell research. How? Stem cell research? Epilepsy? Tell us about that.
Yes, so it's obviously a very exciting new area for research as a research tool, stem cells. And nowadays, you can grow stem cells from just as a tube of blood. It's much easier now. And the stem cells, once the blood cells are converted into stem cells, they can be changed to different cell types of interest. And our interest in the lab, obviously, is around neurons or brain cells. And we use that as in several ways, in particular using as a disease model. So you can use that to model that person's individual's disease, but also using this as a drug screening or drug developing platform or method to screen different drugs. And the uniqueness, I guess, of stem cells is that it retains all the genetic material of that particular individual. And we're all different. Our DNA sequence is all different. But when it comes, so the stem cells would retain all of that information. So if we use that as a way as a model of that particular person's brain or how they will respond to medications, then it becomes a really personalized
medicine.
That's right.
What's the process of this? You literally have some human blood and you're in your lab. What actually happens?
Obviously we explain the research, what it's all about to the person, and they just take a drop of blood, just like when you go to see your GP and you take several mils of blood. And we take it to the laboratory, and then we extract the white blood cells. We remove all the red blood cells, and from the white blood cells, they can then be cultured and then what we call reprogramming. We will program them backwards, if you like, turn them back into stem cells. That process takes several weeks, and we do that in the laboratory. Put them in the incubator as a culture, and after a few weeks, when they're stem cells, then we can treat them as, like in the very embryonic stage. You can turn them into whatever types of cells we're interested in, and that can take for nerve cells, that can take between 4 to 6 weeks, and the cells then becoming active. We can see the activity, what we call firing. We can see spikes when we measure on using electrodes. We can pick up the electrical activities. So it's super exciting, and they're beautiful as well, I must say. They're beautiful to look at under the microscope.
Oh, really? What do they look like, if you can describe that?
You can see strands of nerve cells with strands, how they are connecting to each other. And that really is a model of the brain, because as you know, the brain, our human brain, is a network. All the different brain cells, they are connected in different ways at different cells as well. So, and we can stain the cells with different colors as well. So it's a beautiful, beautiful picture. Almost like the starry sky at night.
It sounds fascinating how you've got these human cells, and you've manipulated them, and they're still around weeks later. Like, they're still, how do you keep them alive for so long?
Well, yeah, you have to, just like people, feed them well. Keep them warm, and you can grow them for weeks or months, and they over time they actually turn into little balls as well. So we call them organoids. Become a 3-dimensional little ball, almost like the size of a grain of rice. So it becomes more and more complex. So we can study them either in a what we call 2-dimensional way. That's just a sheet of nerve cells, or 3-dimensionally, depends on the purpose of the research. And what is even more fascinating, I would say, Torie, is that you remember that the stem cells we reprogram from the original blood cells, those stem cells, we can store them forever. As in, we can freeze them in the freezer, and they're kept forever. So whenever we need to have some more nerve cells to investigate for that individual, we can just thaw them from the freezer and grow them up into nerve cells again. So it becomes almost like an infinite source of materials or cells for us to investigate for that individual.
Do they not become damaged when thawed and frozen and thawed and frozen?
Yeah, they're pretty resilient. Yeah, we have to look after them well, obviously. But
it's like they're pets or something, these human cells.
The mini-me's that live on.
And what particular type of epilepsy are you researching using these cells?
Because these cells keep all the genetic material of that person. So naturally, you would have thought they'd be most relevant for people with the genetic form of epilepsy. And that's some of the focus we have been with this grant, is to recruit people with genetic causes, with known genetic causes of epilepsy. As you know, there are many genetic causes now. Often they disrupt how the signals are transferred or between cells. What we call the ion channels, when there's a firing of the nerve cells, it's because of signal changes. So these are the patients with particular target genetic epilepsy. And because we know the genetic mutation that causes the epilepsy, what we can also do nowadays is with a technique called CRISPR, you might have heard about.
Oh, yeah.
To correct that mutation. So put back a normal sequence of the DNA. So that allows you to compare, and you understand exactly what the mutation has done to the behavior of the nerve cells.
So you're taking a cell from somebody who might have, say, I don't know, Dravet syndrome or something. Then you process it in the way that you've described. Then you have your what would you call it again?
They are called iPSCs, induced pluripotent stem cells, or iPSCs for short.
And then you can edit that cell, can you, to make it seizure free, or you can use it to...
Correct. Yeah, we can edit the cells, so we correct the abnormal DNA sequence. We correct the mutation.
That's pretty swish. That is pretty cool. But this, of course, these are little cells, and they're not... do you use rodents yet, or what stage are you?
Mm-hmm. So the cells, yeah, you're right. I mean, these cells are still just cells cultured. So they obviously they don't represent the whole entire complexity of the brain. But then they are a good platform. This is a good platform because since they're very safe. If we treat animals or even people, whatever intervention we give that may cause harm. So these are cells. We don't have to worry too much about safety, and it allows a lot of high throughput screening and testing. So we do a lot of testing over a short period of time, whereas it will be very difficult even on animals and people, let alone people. And so you use it as a screening test. Once you screen some candidate drugs, for example, then we can take them onto the animals and eventually to the patients.
Do you foresee in a couple of years, in 5-10 years, potentially you furthering this research to go into animals?
Well, that is the goal. So right now we have one of the projects that we receive MRF funding is to use this in vitro or cell system to screen for a new class of drug for epilepsy that currently doesn't exist. And done the screening, then we'll test on animals. And the next stage would be to test in people.
Do you have any numbers, statistics that you can quote in regards to your research here?
Yeah, we were trying to help the 30%, 1/3 of people with drug-resistant epilepsy. That's the main driving our research. 30%, even now in the last 30 years, more than 20 new drugs have been developed. 1/3 of people are still having drug-resistant epilepsy. And developing different methods to tackle that. I've mentioned stem cells is one of the personalized way. The other way we're doing is applying AI to help us select the right medication by learning from large patient databases, how patients responded to existing drugs, and that may help us to this device choose the best medication for that particular individual by using AI.
Oh my goodness, you're just sending my own remaining brain tissue wild in wonder, just thinking about all the potential options of the future. And so it just shows data is so important. So I guess the more of us that can be involved, patients and clinicians, the bigger our data set and the greater the likelihood, fingers crossed, of finding improved treatments for the future.
Yeah, absolutely. In fact, the research communities in epilepsy, we're all very collaborative. We all support each other. And the AI work I mentioned, we set up a international network of centers contributing data for us to train our AI model. So we now actually have data from more than 9 countries, more than 20 centers, and for us to train and test these models. It was really exciting. Data from across for thousands of patients. But the machine is hungry, as they say, the machines are hungry. So also we are very grateful to the patients and who have allowed us to use the data.
And wouldn't it be really useful it's almost essential, I think, to get a greater diversity of patients involved as well, because it tends to be, I'm generalizing, but there tends to be a certain type of person who goes forward for research all the time. But we need to, I think, I have a passion for getting more people from different backgrounds involved. What do you think?
Absolutely, yeah. That is one of the often criticisms for, AI is very narrow and we don't have enough diversity. So absolutely. So we always in our work, that's what we make sure we cover, people from different backgrounds, different socioeconomic settings, from different countries, from different continents, to ensure that diversity. And what we find can be what we call generalizable. It's really can be applied to people with different backgrounds.
Yes, we are all the same species after all, So thank you so very much, Patrick. It's been lovely to meet you finally. And everyone keep an eye on Patrick because he's doing such amazing work. And you'll be presenting at the...
Yes, the American Epilepsy.
ADS, that's it.
Our team will have a few presentations there. So look forward to seeing some of you there.
Thank you so much to Patrick for giving us a glimpse into his exciting research and for making it fun and understandable. Do check out Patrick's papers and more about his work on their website, torierobinson.com, where you can also access the podcast, the video, and the transcription of this episode. And if you haven't already, don't forget to like, comment, and subscribe to the channel. Share this episode with your friends, family, colleagues, and see you next week.